Hello everybody, this is Mr. Halim from Harmony Science Academy in West Houston. Today I'm going to do an experiment about enzymes and vitalism. Very simply, what vitalism is, it's a doctrine that states that living things are different than non-living things because they contain some non-living components and they're governed by different principles than laws of physics and chemistry. That is argued among scientists very long time, but I'm not going to talk about it in too much details. How this idea relates to our experiment is we're going to try to replicate the Eduard BUCHNER's experiment. What Eduard BUCHNER did is, in his time, people used to say that life functions like fermentation cannot be carried out by laws of physics and chemistry. And what he did is, he was working on fermentation of yeast cells. He grind the yeast cells, get an extract from them, having no cells in it, and he used a machine that is invented by him for that purpose, for grinding the yeast cells. He got an extract. He put this extract in the sugar solution, sugar water, and he waited. At the end, he got ethyl alcohol. Actually, what he had in that extract was enzymes, and these enzymes carried out the fermentation, which is relatively a short pathway in an in vitro environment, which means that in the test tubes, not in the cells. Alright, now what are we going to do is we're going to try to replicate this lab. I'm going to use carbon dioxide sensors to detect the carbon dioxide because the fermentation is releasing carbon dioxide gas. I will do yeast extract and sugar solution in one chamber, and I will use sugar solution and the distilled water in the second chamber. If there is no, according to my hypothesis, if there is no carbon dioxide change in the second chamber, that means there is no fermentation is going on here. And if there is, in the chamber 1, probably that indicates that there is fermentation happening here. A further study could be detection of ethyl alcohol in the chamber 1. So what I'll do is, I have my distilled water, boiled distilled water. I have yeast solution made by yeast using yeast extract, and I have 1% glucose solution. I will add 1 milliliters of glucose solution and 1 milliliters of yeast extract in the first chamber. So 1 milliliters. I will open this, add the glucose, seal it, and then get 1 milliliters of yeast extract. And add it. Seal it and mix it. The second one, I will do the same thing with sugar and distilled water. And I'm trying to do close it. I'm trying to close the bottles as soon as I can to prevent any contamination by the air. So I have this. mix it, and leave it here. What I have here is, I have two bottles, two chambers. Chamber 1 have the yeast extract and sugar, and chamber 2 has the sugar and distilled water. And an important point is the carbon dioxide sensor in the chamber 1 needs to be connected in the channel 1 of your interface, which I'm using LabQuest 2 here, and carbon dioxide sensor in the chamber number 2 needs to be connected to channel number 2 in the LabQuest. And after you're done with this setup, you just start data collection by clicking the collect button in the Logger Pro, or you can simply press space button to start the data collection. And it will collect data for you. After your data collection, you can do your data analysis. I will apply a chi-square test for this lab, and it will be in a separate video. So that's pretty much it. See you next time, and thanks for watching Harvard Public Schools.